Hey man, how are you? Okay. Taking your introduction, Stuart, okay? We all set to go? Yeah. Hi, good morning. I'm Dennis Zakowski, Science Director here at the Foundation. I want to welcome everybody to our Edward A. Ames Seminar. And we have here about 10 folks in our office, and we understand we have about 29 people on the webinar. So all of you on the webinar, welcome. Just a couple of sort of housekeeping notes. That those of you on the webinar can hear us, hopefully see us, and be able to respond with written questions later on. And so we'll pop them up for Stuart to take a look at when he's through with his presentation. Today is a kind of a special seminar that we added to the series when we found out from Mark Reese at EPA that Stuart was going to be in town. We don't have your plaque ready for the speaker coming the longest distance ever to be at one of our seminars. We appreciate you being here. But a reminder, our next seminar is going to be next week. We're going to hear about the effects of our past tropical storms and the geology and geochemistry behind some of the changes that took place in the Hudson. So we've got 2 this month. I think we'll see some of you again next week, and we also have a sign-in sheet that if you did not RSVP to the seminar, we just ask you to sign in so we can keep track of you and make sure that we're contacting you for future events as well. So I'd like to ask Mark Reese from Region 2 of EPA to introduce our speaker.
Yeah, it's my pleasure to introduce Dr. Simpson, and I'm very excited to hear what you have to say. I've been a sediment manager in New York Harbor for 20 years. I came in and started at the Corps, and I've been at the EPA for 18 of the last 20 years. And coming in from graduate school into the job that was available at the time as a sediment manager, I had a history and ecology background. So I jumped in to get up to speed as quickly as possible on, how was I going to manage sediment in New York Harbor and make decisions in light of the varying quality that's there. And at that time, EPA was pushing development of sediment quality criteria, which would have just been perfect to make decisions with, on individual contaminant in sediment, just measuring sediment, and away we go. It sounded so simple and perfect. There was competing efforts by NOAA and state and others. So, quickly it became clear that there was sort of an alphabet soup out there that contaminated the sediment quality criteria, ERLs, and ERMs, and AETs, and PELs, and PELs, and et cetera, and there was empirical guidelines and mechanistic guidelines, and there was consensus guidelines and quotients and normalizations and all kinds of things to contend with, all in search of the holy grail of can we come to a number that we all agree on is, can differentiate between good quality and bad quality. And of course, it wasn't quite that simple. There's the other contaminants in the mixtures to contend with. There's the geochemistry. There's how did you sample? What are your assumptions? So it was all very complicated. And being at the Corps at the time, I was very well trained that in light of all those uncertainties, we simply can't make decisions based on sediment numbers itself, and we would invest ourselves wholly in toxicity tests and biological assays. And so you had these 2 camps proceeding for most of the 20 years I've been working, pretty much without recognizing the value and the weaknesses that each of those approaches brings to the table. So when I found out that Stuart was, and in the last 10 years, I think there's been reconciliation of that tradition, and there's been recognition that, that these are lines of evidence and that we need to work them together to really come up with, responsible management decisions based on sediment quality. So when I found out that Stuart was available to speak, I was very excited, particularly after he shared what he wanted to speak about, which is, in fact, how do we make better decisions using all the tools that are available for us. And so Dr. Simpson comes to us from Australia. He's got 20 years of experience. I'm sorry, I'm going to have to read some of this. He's the senior principal research scientist and leader of the aquatic contaminants group at the Commonwealth Scientific and Industrial Research Organization, which I guess is equivalent to the, would you say, to the NSF here, or is that?
I don't know what that is. I'll talk about it soon.
Okay, great. So, with no further delay, thank you very much for coming.
Yeah, thank you, and thanks for hosting me here. It's a great institute as well. Thanks. I'm going to have a few short overview slides of CSIRO just to start off, so you've got an idea of what we are. CSIRO is a federally funded research organisation, scientific industrial research, across on a very broad scale across Australia. But at the same time, we have to bring in half our money externally, so we do a lot of contract research. We work very much at the interface between industry and government in many of these areas. We're not making regulations, we don't regulate like the EPA, but we try to provide science, at that interface and provide the right tools for industries to get on with their job of their industries and government, on regulating at the same time. CSIRO is about 5,000 people, probably about 7,000 people 5 years ago. Perhaps 4,000 in a few more years, the general scale of scientific research and government, spread right across the country around many sites. I'm based in Sydney. Sydney's a big harbour city, deep harbour city like New York, beautiful harbour. We don't bridge and we don't have boats going in and out either anymore. The big ships, as they go off to our little Newcastle and Wollongong either side to get them out of the harbour, which is becoming more and more about people these days and things. But yeah, so CSIRO has a whole range of business units, they're called these days. A few years ago, they were flagships. But right across the board, agriculture, health and security, data, energy, manufacturing. I'm based in land and water, it's my home, but I do a lot of work in oceans and atmosphere, which brings me more into the coastal and even offshore oil and gas world of things, and a lot of work in our mineral resources world. Our mining companies produce a lot of contaminants flow from industry. It's where they, often start off lots of things we're digging up and sucking out of the ground and things. I manage quite a small group in Sydney within another program in land and water, and it's sort of quite an integrated group of chemistry to toxicology. I say omics these days because our ecology, the one slide at the end, is really transformed into genomics these days as we've moved away from some of our traditional ecological methods in some respects. The issue I'm dealing with in many ways is there's billions of dollars we spend, globally, nationally, on assessment, measurement, assessment, regulation of contaminants in waters and sediments. And if you can do that more effectively, one, it costs less money for industry and for government. Government spend a lot of time arguing about things that maybe they don't need to argue if they had the right tools, or decisions could be made more easily. But also we could protect the environment and have a great conservative with which we protect the environment. And so, make the comment that inadequacies in tools, guidelines, frameworks, all that impede our decision-making processes. And that's largely the essence of the talk that I'm talking about today. The general title is Advancing Guidelines and Methods for Sediment Quality Risk Assessment, and a lot about incorporating bioavailability. While bioavailability has been talked about a long time, and it's written into most larger assessment frameworks and guidances, often it's more talked about than people actually doing things, and some of it relates to misunderstanding of the appropriate tools, or they're just not being the appropriate tool, and so I'm going to talk a lot about that. I mean, sediments are very complex. There are a lot of different organisms within our sediment environment, doing a range of different things. Some are just living in the sediments as their home and living off what comes out of the alkaline water. Some are head down deposit feeders, ingesting everything. Some are very selective. than what they ingest and eat. So they've got a range of exposure routes to contaminants. At the same time, contaminants are stratified with oxic surface layers, oxic down the burrows, with oxygen penetrating, going suboxic into very deep and oxic sediments, where things are bound very differently and the bioavailability changes. It's stratified, but it's also dynamic. Sediments are moving, particularly in harbours with big boats and turbulence, or in coastal environments. This is Australia's framework for sediment quality assessment. In the year 2000, we introduced sediment quality guidelines, and being a much smaller country than the United States, it's much easier to go, right, we're going to have guidelines, and someone's got to make a decision about them, let's just get them in place and things. And we went about it by having a tiered decision tree approach to how we're going to, assess the risks of contaminants and what hazard they pose to the environment. We have a decision tree, and this is just the one for metals. We have a separate one for organics to some degree. But basically, the start-stop vote, we're looking at total concentrations of contaminants. Initially, you get a feeling for what's out there. And at the same time, we think about the sediment properties and other factors and so on. But, they are just guideline concentrations, and part of my talk is going to be about the value of having guidelines. There is a value in definitely having some sort of guideline. It doesn't have to be a sort of criteria, but you need some sort of starting point where people start thinking about there is a risk, or there's really no risk at all, or there's potentially a risk. And then the major part of my talk is about bioavailability, because once you exceed some sort of idea that these contaminant concentrations are high, they're elevated, we need to understand whether they're bioavailable or not, and how that information flows into our decision-making processes. We decide there's bioavailable contaminants or potentially bioavailable contaminants. We step through in our guidelines doing toxicity testing, look at a whole range of other lines of evidence, like the bioaccumulation, bioaccumulating substances, mercury, the organics, and then other broader lines of evidence in ecology. I'll touch briefly on ecology at the end. It still sits off the side of many of our assessment processes, the way it generally works. At the same time, I also note that many of our contaminants, by far the most, probably 99%, don't have guidelines, and how important is that? There's lots of them, but are they the major contaminants? During my sort of career, I came in as a chemist, so not coming in as the fish biologist or that sort of thing. I came in as a chemist, but you've got to start asking questions about organisms, and more so in my career, I've become a biologist, a toxicologist over time. And as I look through our decision tree process, more and more you delve into it. There's lots of questions associated with each of these components. Do we have any guidelines in place for many of these contaminants? What are the useful methods for doing bioavailability assessment, and so on? What effect measures do we have? Are they appropriate? The toxicity tests we use, and so on. Sediment quality guidelines are, very much less reliable and have much greater uncertainty than many of our water quality guidelines. And water quality guidelines in the derivation process in most countries and jurisdictions that follow to derive them is your dosing waters with different properties with a single contaminant and looking at the effects to a whole range of species. And you might get a case like this. This is copper. There's lots of data for copper in both on freshwaters and marines. These are all no effect concentrations. So this organism up here, the little market's disappearing. It's affected at a very, very low, at a high concentration. These organisms down the bottom here are affected at a very low concentration. You can look at a distribution of them and go, you can make some sort of calculation of what concentration you might need to protect 95% of all species with some degree of certainty. However, that's copper. Many of the contaminants, chemicals in the environment, we don't have much data on. In the case of sediment guidelines, they've all come through from these data sets where we've got everything there present all at once. Some people have done some spike sediments, but with a single contaminant, but most of the data is coming through from assessing the toxicity of sediment and looking at all the chemicals that are there and trying to attribute the toxicity to an individual contaminant. The issue becomes, it becomes quite easy to say when you're not going to have any toxicity. Generally, it's down in the background concentrations, and it's actually quite easy to say when you're definitely going to see effects too, and that get very, very high concentrations often. But there's a huge grey area in the middle, and this is, really all due to bioavailability and variability in the mate species and the combination of the chemicals that are present in the sediments. So I've talked a lot about this and done a lot of my own personal research into guidelines for copper in my career, and not really due to mining industries behaving badly in that sort of world and things. But guidelines, what are they based on? Where's the concentration response relationship? And this is effect status. This is survival data that we're seeing here for an antipod, which we use quite a lot. It's survival data. And this, as we can see, as we go up in copper concentration, we can have sediments that have thousands of milligrams per kilogram of copper and have no effect on survival. But at the same time, we can have the same concentration, have complete annihilation of this organism and wipe out. And that goes across the whole span of concentrations. It's only down at the very, very low concentrations where you get sediments where you don't see any effects to this organism at all. So how, if you go out there in the environment, you take a sample and you measure concentrations, you go, is this a high or low risk or not? You can't use total concentrations in that sense to make a decision on the risk of effects and risk of toxicity to an individual, let alone populations of organisms. And this sort of flows right into our this big grey area of uncertainty. I mentioned that bioavailability and guidelines the guidelines are much less certain and much less reliable in sediments. And the reason for this is metal bioavailability is much more complex than it is in waters, and much more variable in many respects as well. In waters, we deal with some issues of bioavailability. You've got metals or organics that are complex, partitioned to particles that are in the water column, and there is multiple exposure routes. We don't deal very well with colloidal particles, so I really understand how bioavailable they are, but they're obviously less bioavailable than the dissolved phase and dissolved concentration. Sediment, there's a lot more going on. We've still got to deal with what's happening in the overlying water, because organisms are sourcing contaminants from the overlying water as well. But we've got varying bioavailabilities of contaminants, and here we're looking at metals in the sediments, potentially more bioavailable in surface sediments than when they're bound in sulfide phases in the deeper sediments. But ideally, our guidelines, whatever we put in place, should really be concerned with that bioavailable fraction, concerned with that viable concentration. At the same time, realising that total concentrations can potentially transform into more bioavailable forms with time. So back onto the broader part of my talk now, it's about bioavailability and how do we bring this into our assessments, how do we use it properly, and what benefit it doesn't have to our decision-making processes. All the work I've done over the years is, you look at a tool and you're trying to work out how you use it, and they often work well in some situations, not others. And in general, I found there's no single tool that's effective for assessing bioavailability of metals in sediments. And the same applies to organics. There's no one tool. There's a range of sort of chemical extractions people have proposed. Say this captures the bioavailable fraction, excludes the more inert fractions of the contaminants. Equilibrium partitioning calculations, which are really trying to calculate how much might go into the water, into the dissolved phase. Works with varying success. The acid volatile sulfide model we'll talk about with metals. It's very useful in some cases, but it's not without its faults and flaws. Many organic contaminants position strongly to organic carbon, but how do you deal with different forms of organic carbon? There's lots of forms of organic carbon in sediments, and they have different binding properties for organics as well. Forwarders, very useful for measuring, the bioavailability in that phase. But once again, many times they provide just a snapshot of when you measure them, and is it the best predictor of effects. I think the battery's running out of this. Yeah, I wonder if you go down. Yeah. So metal bioavailability sediments. I mentioned the ABFSM model. So acid volatile sulfide. So sulfide binds these metals up here very strongly, or puts them in forms that are in a less toxic form, in the case of chromium. And if you've got an excess of this form of sulfide, acid volatile sulfide, over metals that are extracted under the same conditions, basically they're going to be bound in sulfide phase, not partitioned to the water, and be less bioavailable than if they were potentially partitioned to more oxidized phases. And there's been a huge amount of work done in this method. And basically, it captures some of the uncertainty in this area down from a small amount where you've got this big window of when does toxicity really occur, down to a smaller range when you take into account the binding of these metals to sulfides in the sediments. It doesn't necessarily deal with metals in surface sediments very much. And one of the issues with ABSECM2 measurements is the fact you can actually make a measurement in the bulk sediments and make a prediction that your metals will be bound up in that phase, but many of your organisms are modifying the environment directly around them. So the bulk sediment properties aren't necessarily predictive of the effects they're seeing. So we've been trying to deal with this, and this is once again back to the case of copper. And we had some very good students and researchers over the years doing a lot of work on dosing up sediments with a whole range of properties and looking at how do you predict the toxicity of copper in these sediments. And this is effects to reproduction, and you've got a log scale, so you can see these concentrations in which this sediment causes toxicity versus another one causes toxicity are over an order of magnitude or larger in many cases. And we're particularly looking at the effects of organic carbon and binding of metals to silt in these sediments. And what we were able to show in the case of copper was the fact that if you normalise your concentration, the amount of copper in the fine fraction, and to the amount of organic carbon in the sediments, all of these concentration response relationships, or these dose responses, collapse into largely a single relationship, and have a much better ability to predict the toxicity to this organism. So, by bringing in this understanding of how the sediment properties influence the bioavailability of copper, we've got a much better ability to predict effects. So, part of this, how we're using it and how we sort of propose to use it in Australia in guidelines. Well, it is sandy sediments, low sulfide, low organic carbon. You've got less ability to bind metals. Metals present in them are going to be more bioavailable compared to as you shift towards more silty sediments, high sulfide, high organic carbon. And in theory, if you had species sensitivity distributions, your distribution would be lower down. You get a lower guideline concentration being derived for a sandy sediment than you would for a silty sediment. And theoretically, you can predict how this would go if we have enough information on different species. Is that a sediment with high amounts of silt, high amounts of organic carbon could have 500 milligrams per kilogram of copper, and it'll be bound up and be not bioavailable to virtually all species, versus having sandy sediments which have low organic matter, whereas really you have to have a guideline concentration that's much lower. There's problems with this approach though. A lot of time going, hey, this is going to be really good. We're going to derive guidelines that we can modify based on sediment properties, and it's going to save a lot of problems with our assessments. But it does help with decision-making processes and helps with those discussions around whether something's bioavailable or whether there's a risk or not. The issue becomes is that colleagues are working with doing the same approach with nickel and freshwater sediments. And the problem is there's different binding phases that really control the bioavailability of nickel, and it would be the same for cadmium and zinc, something else. They've all got different binding phases that modifies their bioavailability. So you get to the issue of there's going to be no one universal guideline on modifying tool in this case. ABSECM does part of that, but it doesn't do it that well in all cases. So, with my science been going? So, for some time ago, we had Bill Davison, who developed this technique of diffusive gradients in thin films. It's a really technique developed by for soils, but really for waters, and trying to capture, the concentration, a time-integrated concentration of metals in the water column as they diffuse into this passive sampling device. That can also provide information on bioavailability, from those measurements as well. But I sort of, thought about this, over many years, but I suppose it didn't really occur to me how I could apply it to sediments until I saw this paper where, they showed these beautiful profiles of metals and sediments. And you just need to think of these colour intensities as this is a concentration of flux. Don't worry about what the scale is. You see this is depth down into the sediments. So we have sediment water interface, and then, wow, right where this little epibenthic organism that I've been using for toxicity test lives, there's a huge flux of copper. And therefore, if you make a measurement in the bulk sediments, or you make a measurement down below water, what use is that for predicting the toxicities of this organism? Can't see how it's much use at all to some degree. And then you've got the issue of different metals have different profiles and where the highest intensity of metal release occurs. And you've got organisms living within these different areas within the sediments. Part one. The other part is that, metal fluxes uptake rates and how it relates to toxicity. Sort of following the research by Phil Rainbow and Sam Law over many years, has done a lot of work on modelling exposure routes and dissolving dietary routes and so on. And basically, what you come up with from those models is, toxicity is going to condense when you're overwhelmed with some flux of metals and contaminants in your body. Same with us as humans. You can drink alcohol slowly, your body can process it, but if you drink it too fast, you're overwhelmed and toxicity occurs. So you have that uptake rate, the flux into the organism. It's not just the combined total concentration, which is why bioaccumulated concentrations of metals in field-collected organisms isn't that useful for assessments either in some respects. The other aspect of it is we want the uptake fluxes and rates, but we also need this over time. It's no just having a snapshot in time and going, we're going to make an assessment based on one measurement in one point of time or one sediment type. You want some sort of integrated time average measure of the concentration or flux, and in theory it should be more useful for predicting effects to organisms which are exposed for long durations. So moving on, I had a very good postdoc from Europe who came and worked with me. We pursued on with this, how do you predict toxicity in sediments? And now this is a pachycoid copepod that lives in the surface layers of sediments, doesn't burrow very deeply. And once again, we've got sediments that are dosed with copper, whole range of concentrations, different properties as such. How do you predict the effects to the organism? You can't do it based on the bulk concentration. It varies sediment properties. But you could, once again, if you normalise to the amount of copper with a fine fraction in organic carbon. But also, we had a biokinetic model for this organism in the amphipod. We're able to predict the exposure to this organism, and this exposure can both be dissolved in the dietary routes, in this case coming from the organism. And if you understood this uptake rate, it's not the overall net exposure, but the uptake rate in this case. So it's micrograms of copper going into this organism per day or per hour. If you've got an idea of the rate of exposure, you have a much better ability to predict the toxicity than, once again, total concentrations. And other people working on how to use time average concentrations, largely water column effects, pulse discharges into a system, how do you use that and compare it to a guideline, that's a chronic guideline, and doing work on this. And basically, in the case of metals, and a lot more follow-up work on this, if you can use a time average concentration, it's much better at predicting effects regardless of what the exposure is. You can have all these pulse exposures of high concentrations or longer exposures of lesser concentrations or different exposures to this organism during a bioassay. But you're able to predict the effects based on a time average concentration. So, let's try to bring these things together. Okay, we've got this tool that people have developed, diffusive gradients and thin film, sort of talk about now. It gives you some sort of flux of contaminants with a passive sampler for measuring the flux. And it integrates over time. How do we use it to predict toxicity? It's not being used, most people using it. It's more about geochemical studies and using it for different purposes. And I want to say, can we use it for the purpose of risk assessment? So this is the device. Basically, a plate, and then you've got a binding layer that binds cationic metals. There's a range of binding substances that can bind different things, but this one's called cations, cadmium, copper, nickel, lead, zinc, those sorts of cations. There's a diffusive gel, and there's a membrane, and there's a plate. And you can put these into waters or into sediment in this case. The metals diffuse into the gel, and they get bound at the resin, go to zero concentration, and they keep diffusing in. And so basically this device is just accumulating metals with time. In the water column, you can back calculate, given the diffusion coefficients, to calculate a concentration. In sediment, it's a real mess because you've got supply issues that you deplete pore water of the concentration and so on. So I was going, well, let's forget that. I just want a measurement that gives me a flux, gives me a concentration. If the sediment's releasing a lot of metals, it should be a higher risk to organisms and the environment than the sediment that isn't releasing a lot of metals in this way. And so that's how I want to use it. You can also slice things at different depths. So you slice them at the sediment water interface, or in the surface, overlying water, or deeper down in the sediments. In what part of this life, and where should I make this measurement, and so on. So I'm going to talk through about 5 case studies quite quickly now where we've been applying this method. This was a study, a laboratory-based study, but it was based on a real issue. Our Tasmanian aquaculture industry for salmon has these huge nets of salmon. They're probably about twice the size, 4 times, probably 4 times the area of this room we're in now. They're huge. And they were painting their nets with copper-based anti-fouling paints. Once not biofouling, I go, why copper? You know, it's the most toxic metal after you've got rid of tributyltin, now using copper. They go, well, it's a stiffer paint than zinc. Well, you know, because they wanted stiff nets, the seals come, these fellas come racing in, hit the nets as hard as they can because they want to get a big bite of salmon, even if there is 2 nets. So there are practical issues with that. But the issue was, you know, the paint's meant to diffuse and allow copper to go off and stop biofouling. Most of the paint lands in the sediments, and that's where it ends up under these agricultural issues. Thankfully, due to our research and other global pressures for, you know, sustainable food production, things like that, they're out of these types of nets now and using other types of ones that require more work that don't contaminate the environment so much. But, so we were dosing these sediments up with copper once again, copper-based paint this time. Copper paint's not very bioavailable at all. It's pretty, try to dissolve it in dilute acid, you know, which is very strong compared to what an organism would have to extract it. You don't get any copper out of it, very small amount of copper. So it's its original state, it's not very bioavailable. It's meant to diffuse that slowly. So looking at total concentrations, we have these different dose-response relationships for sandy sediments going up to silty sediments. You know, well, you measure that concentration, what risk would you predict? We measured dilute acid extractable copper, which is a 1 mole HCl extraction. You know, it's meant to give you a better idea of the bioavailability too. And probably in many of the studies we've done, it does a pretty good job at it as well. But you go, well, there's not much different. There's no better, no improved predictability of the toxicity survival of these bivalves in this case by taking that step of saying, well, that's a single extraction that might give you some information on bioavailability. We applied our DGTs to a whole range of sediments in the laboratory where we had these exposures. And this is what we got as far as profile. So the silty sediment, copper's bound up quite tightly in these cases, but at the highest concentration of paint associated copper in the sediment, you get a little peak right at the sediment interface. And you move down to the more sandy sediments, your fluxes get higher and higher. The copper hasn't been bound, because copper is more bioavailable, so there's many binding phases there. But it certainly showed what we thought. Well, right at this interface, there's an exposure of copper to this organism. I won't show you all the other figures, but basically we're able to use this flux of copper. So it's not a concentration, it's just a flux out of the sediments to the DGT. So it's the device, passive sampler induced flux to some degree, and accumulated. Regardless of the properties of sediments, we're able to predict the effect. So, you know, we're going, well, it could be quite useful. Above a flux in this slide on this case of 10 micrograms of copper per metre squared per hour, it's just something you measure off from a device. There becomes a high risk of adverse effects to the survival of this organism. Two very good French internship students who worked on this over some time up there in the top corner. Okay, well, that was just copper. Simple. Studied copper a lot of my life and it's pretty toxic. What about metal mixtures? Sydney Harbour, like out here in front of us, so a legacy of big metal industries, you know, discharging things over 100 years straight off into Sydney Harbour. So we've got lots of sites, and we've got huge amounts of urban runoff. So you've always got zinc and copper and lead coming in from roads and stormwater over the years. So we've got some very contaminated sediments in some cases. And so I wanted to look at these. You've got more complex mixtures now. Your metals, you know, your copper, lead and zinc as the dominant contaminants, but other things are present as well. Can we predict the toxicity? And so moving on from survival and reproduction of the amphipoid. This has 2 points for assessments here, is the fact, you know, often people have developed toxicity tests over time and go, right, we've got a 10-day lethality test. Has been developed, standardised, and people just start using it and don't look to develop a better test, a reproduction test, if it's too difficult or has a few issues, or the fact that this other test has just been standardised. Survival very sensitive compared to reproduction, and we're trying to protect against, whether our organisms can reproduce these sediments and sustain themselves. And so these sediments, this is with an order of being more contaminated. We had effects to survival in the very most contaminated sediments, but all of them had effects to reproduction, and some were complete wipeout reproduction, and some were just very large scale effects to the reproduction of the samplepod in a test. But once again, we're able to use the flux and the combined flux of copper, lead and zinc in this case to show, well, we've got a nice dose-response relationship, and in this case, we need to think about, well, how do you normalise the fluxes of different contaminants, copper, lead and zinc, and because they have different toxicity to predict them. And in this case, we went through a range of approaches where we're going to divide by the effects concentration specific to this organism, or just divide the flux number by the guideline, the water quality guideline going into these. And that's what we did in the end. So these fluxes are the flux of copper divided by its guidelines, 1.3 in Australia's case, copper 4.2, I think, zinc 8, and divided by that, and then summed together. So it's sort of a combined effect. But here we've got above, once again, in this case, a flux of about 3.5 micrograms per gram combined with these metals normalised. You're seeing large effects to survival, and maybe above 1. In this case, you're starting to see effects to reproduction. So again, we're getting to the stage of, you take a device like a DGT, put it in sediments, and measure a flux above this number, it's going to have an impact on these organisms and certainly other organisms in the environment. This is a step forward as for use of in situ passive sampling of bioavailable metals. Well, that was all laboratory work when we get out there in the field. So we had experiments we were comparing between field and laboratory evaluations at sediments, naturally contaminated sediments, a whole range of metals, and this is bioaccumulation in this case, by bivalve in the field, in sort of mesocosms and containers in the field, and in the laboratory. One of the things for assessments, and we're dealing with the industry and the government, they need fairly simple tools, and it's always been in my mind that is DGT, are all the passive sampling tools, are they too complex? Is a consultancy, who's going to be doing this in many cases for industry, and they're going to grab a new graduate from the university because they're cheap, or from somewhere else, and go, get out there, fit this program in the environment, pull it back out 24 hours, come back, send it to this lab, and give us the number, and we're going to use it for our report. That's quite complex, rather than going out and collecting a sediment and sending it off for analysis. And so are these devices providing a measurement that's much better than something that's simpler? So that's one of the questions here. Again, this is accumulation. You go, well, this is the flux of copper and flux of zinc, and accumulation by this bivalve. And you go, well, above a certain flux, you're getting a much higher level of accumulation, whatever this accumulation is in our assessment anyway. But is it any better than what you would have predicted based on a measurement as simple as low acid extractable metals, 1 mole HCl extractable metals? In this case, you go, well, maybe there's no benefit. There is a real benefit of having the in situ method in this case. Maybe not. We're also applying these in freshwater environments. Most of my work's coastal marine, but also in the freshwater sediments. This is downstream of a historic mining area, which had a lot of metal contamination, but largely zinc was the main contamination in this case. And we're looking at accumulation by bivalve again. I look at these profiles. These are profiles from the DGT and the sediment. So each one represents a concentration averaged over this space. And the other thing, you sort of think, well, how on earth can you use this information to predict effects and things? Looks like a mess to me. Can't be very useful, but what we'll find is it tends to be quite useful. I'm just showing it here for zinc, but useful for, in that case, cadmium, which also has quite a high concentration. But we're asking the question here as well of what depth should we slice this device, because this bivalve corbicular one was buried quite deep in the sediment, and should we be using this slice at the sediment water interface as the exposure rate? Many bivalves actually have their siphon out and siphon around and sucking that detritus off the surface, so they might live down there and get some exposure down there, but most of their feeding is up there at the sediment water interface and so on. But here, so these are, once again, DGT fluxes, and using them to try to predict the bioaccumulation in this case. And this was in the laboratory and the field, so regardless of whether it was the laboratory or the field, they all sat on the same line of flux predicting the exposure. The ones in the laboratory are the ones with the higher flux. The water isn't being flushed away in the testing chambers as readily as it is in the field exposures in this case. But in fact, it didn't actually matter whether we used the flux in the overlying water or the sediment water interface or the whole sediment. The relationships were similar. Maybe once again the sediment water interface was better, but we're sort of thinking about that issue as we're going to say, well, we think the tool's useful. We're going to provide practical guidance on where to make these measurements. Is it better than the other tools? So this is the same experiment. Well, this is our DGT flux and zinc, and this is our relationship of bioaccumulation with total particulate metal concentrations, no relationship at all. And with dilute acid extract metal concentrations, really no relationship at all. So you go, well, in this case, it looks like it's pretty good. In the other case, it wasn't any better, but it wasn't any worse. Here, it's certainly a lot better at predicting, the bivalve metals and what's available to accumulate by this bivalve. I'm going to move on more rapidly now to these last ones. This is quite a complex experiment dealing with, should you have one or two species in a test chamber. We do our toxicity tests where we've got single species, many of them, in a little isolated environment, but in the real world there's all sorts of animals there churning up sediment, doing different things, and trying to understand how that influences our outcomes of our toxicity tests. I'll give you the outcome of that. Is for the case of metals, it generally means you remove more metals and they become less toxic when you've got more organisms mixing around and doing things. In the case of hydrocarbon contaminated sediments, they generally become more toxic. So it does change your outcomes. So the way you do tests certainly influences things. But here we're putting our diffusive gradient thin films into these sediments. We either have just our one test organism or two organisms in the sediments. Don't really have to worry about that aspect of the sediment, but you've got a whole range of these profiles. Once again, in this case, copper, lead and zinc, flux and accumulation by this bivalve, much better relationship than we had with dilute acid extractable metals or with total particular metals. It was useless, and it's showing you that. But seems some value in having this passive sampler for assessing the particular bioavailability and potentially providing a more predictive tool for assessing the risk of these sediments than some of the other tools we use, and it is giving some time integration. So summing up this talk now, part of what I was interested in doing is, I was looking at our decision tree in Australia that we use for doing assessments, and we measure total concentrations, and if you're above some guideline trigger value, you go, right, you need to understand whether this is really a risk or not, and work out how you're going to deal with it. And then it steps to bioavailability, and all the consultants come running to me and go, what am I going to use? What's the tool? And there is no one tool, and there's no one best tool. I go, can we find a tool that fills the gap and works universally on the side of metals? There's a lot of tools for organics now. Rob Burgess, EPA, Organics, provide a lot of advice on that, and they're going very well, I'd say, those tools in many cases. But metals would be sort of lacking this tool for sediments. And so this is what my mind is hoping to get, toxic curves, flux of top metals, contaminants to the organism, specific organs in the organism in many cases. And we want to have some time integration of that flux. And if that flux is high, we're probably going to see toxicity, but in our case, it would trigger you to go on and do toxicity testing and do ecological assessments as well to help really make decisions on these sort of things. Based on my understanding now of DGT as a tool, I think it can improve our suite of tools for decision making and assessing bioavailability. That's one of the key questions, though, will it become a preferred tool? You've got to balance up the complexity of the measurement technique that people are making. You've also got to have these things, you've got to be free of contamination, you've got to put them out there in the field, and you've got to collect them again. Might be useful for some assessments, but can they be still replaced by some of the less specific tools, potentially in many cases too. So I think there's still more work to be done, but I think it's well on the way to being providing a useful line of evidence in passive sampling. That's really the end of this main part of the talk, but I just want to skim over a few other things relating to broader assessments in general and things that we've been thinking about and doing a lot in Australia in many areas. When we introduced our sediment quality guidelines in Australia in 2000, there were no whole sediment toxicity tests. So we said, right, here's your guidelines, here's the decision tree, go ahead. They go, what test am I going to use? Regular tests. And some people had just started using one amphipod toxicity test, and we thought, well, how appropriate is it? And we looked at it compared to about 10 other amphipods, and went, well, it's the least sensitive out of all these organisms. And so those sediments that were tested and being safe and dumped in the ocean probably shouldn't have been in that case. So, we spent a lot of work trying to develop, better tests in many cases. But one of the issues always faces, and get it from industry and consultants to work at that interface with them, they go, we want to do this assessment, we want the answer, we really want it last week. The clients want the information, they want to put this marina in, they want to dredge here and do this. And they don't want to wait 42 days for a tox test and then another phase of that, and 2 years later the finances are gone and the whole thing didn't happen and so on. So, it seems, you're always balancing up the issue of, what, and government needs to make those decisions as well. They're only pressure to make, those decisions in Australia and many areas in our state, certainly so. And so, you want to be able to provide rapid information that's both sensitive, provides a sensitive and robust endpoint. And to me, it wants to be sublethal rather than lethal. I say sublethal means, things keel over and die. We can always predict that in many cases from some of our guideline concentrations. But, when do we start seeing effects to reproduction, which really means the organism can't survive in that environment either, doing that die with lethality. So we've been doing a lot of work on developing new whole sediment toxicity tests that provide that information, but quite rapidly. This isn't exactly the test we do now, but it's a 10-day test where we're getting out effects to reproduction of a hepatopoid copepod. We can do the same thing with our amphipod within 10 days. You're not looking at the whole life cycle, but you can get something that gives you that information about effects, the sublethal effects rather than the acute effects. And this is a quotient of a whole range of contaminants. But also the dose response occurs, similar to where our guidelines are, and we've done a lot more work on this, that these organisms are quite sensitive to metals and organic contaminants. And I spent a lot of my career criticising our guideline concentrations for sediment contaminants. So to some degree, together, they're actually not too bad. When you look at them all together, they're in about the right spot of where they should be as far as numbers, as far as when organisms start seeing effects. So that's toxicology. We want tools that can give us that information more rapidly, whenever we test greater numbers of samples, but sublethal effects. The other part is, it's always troubled us with our assessment. Ecology is always set off the side. You look at your chemistry, toxicology, and you try to develop dose-response relationships. Sometimes you can, they're quite nice. And then someone says, what about the ecology? You measure, you go reference sites, and what's a reference site? You try to find some things you're going to call reference sites. And you look at your ecology and your contaminated sites, and well, they're different. They're different species, there's this happening. And you can't bring out dose-response relationships very easily. And it becomes very hard to make decisions. And part of that relates to just the fact you've got so many organisms, they're doing different things, but also it's the ecological methods. The morphological methods, you collect the sediment and sieve it, you try to identify under the microscope the organisms you can. You normally get 50 or 60 or 100 different organisms you can identify to species or genus level. But is that enough? I mean, there's tens of thousands, hundreds of thousands of organisms and species out there and things. So and it's time consuming. People go grey and nuts, under the microscope trying to do this, and that still base, we're losing it quite quickly. We're going, well, how do we use our modern techniques with DNA, RNA, genomics to make this process more effective, more holistic, looking at everything? And so this here is data for the whole of the estuary. It's about 100 kilometres along the New South Wales coast, where Sydney Harbour is the contamination, which is Port Hacking, where we got it from here. Port Jackson, sorry, here in the middle. Port Kembla's down south, the industrial ports where all our shipping goes into, and the Hunter River where all our coal used to go out of. Until coal was no good. Still a lot of coal being sold. And then estuaries that are largely pristine, right down the coast. And we're looking at, this is this is diversity of organisms in this case, and each of these are sets of estuaries. The points are different estuaries on the morphological side, and this is the data we've got species identified under the microscope. And then we've used, for the same sediments, molecular techniques, take the sediments, extract the DNA, blast it across these databases and go, right, we've got all these operationally defined taxonomic units of species. But what we're able to show is you get much greater separation and better separation, in this case, by using these more modern techniques to go, well, these estuaries are clearly much different due to these ones, and it's due to these types of organisms that are driving the differences. And therefore, it's take that information and push it against environmental variables and contaminants. You can, pick out tipping points for species and differences much more easily, in my view, than you could with the molecular techniques. Going on, this is probably 2 or 3 years ago now, one technique doesn't wipe out the other. They're complementary techniques. You still need to be able to identify organisms and know exactly what they are. But I'm quite confident, not coming from this ecology background either, that, I think 10, 15, 20 years from now, these are the techniques we're going to be using, and they're going to take over many of our traditional techniques in that world. That was just one aspect of it. That's the DNA saying, I'm telling you that that's a human, that's a pig, that's a antipod, that's a copepod, that's a bivalve. It tells you the diversity of species. We're slowly working out how we can do abundance of certain species, but it's a bit more tricky than just counting the numbers of organisms. But you do get all the functional genes, and we're using our human health side of things too. For example, it tells you, what's happening. So we're picking up all the markers of, you're getting, it looks like these sediments are processing nitrogen properly. All these nitrogen functions are happening properly, so the bacteria must be doing the right thing in these sediments. There's all the gene expressions that are telling us, different organisms are doing these things in the right way. It also allows for sort of a next generation of toxic identification evaluation, which is where we're going, this sediment's toxic to this organism, what's causing it. And if you know that dioxins, PCBs cause this sort of gene expression and change in these organisms to go up or down, and metals do something differently, but it's a different gene. In the future, and we've started doing this in some of our organisms, you're going to be able to use these profiles, expressions to go, well, you're targeting the wrong industry. You're trying to stop these metal contaminating industries over here. You should be focusing your efforts on here. We actually know it's not about toxins, it's about the nutrients and all that flowing through the trees. It's a bigger problem. If you want to get a healthy system, you need to be focusing on that. So, there's going to be a lot of information coming out of our transcriptomics, our molecular biomarker approaches, into assessments, I feel, in the future as well. This here was actually a study with our antipod. This antipod eats a lot of sediments. Gobble, 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 sediment interface. It's trying to get algae and bacteria to try this for food, but it eats sediments as well. And we've got this one I've got a nice biokinetic model for uptake, radio tracer uptake studies. We're able to create these exposures in sand where we had high and low dissolved copper concentrations versus going through to ones where we've got exposure coming predominantly from the dietary stage as well. And what I will see from using these using transcriptomics is you can get very clear changes in what's happening to the organism internally, whether the exposure is coming from dissolved or the dietary phase. How do we use that information in systems? I'm not quite sure at the moment, but I'm confident that we'll sort some of those things out in the future. Yeah, I'd like to thank some of my colleagues. Graham Batley's been my sort of senior mentor throughout the vast majority and still my career. He works 7 days a week and we pay him one these days. One of those scientists who can't retire. They're quite good to have. Always had lots of good people in the laboratories and good collaborators or good students. I haven't shown them all, but there's lots of good people I collaborate with, both internationally and around Australia. So, thank you.
Questions? I was very interested by what you said on the blood the molecular versus morphological. Typically, we're not doing anything on that, and it certainly seems interesting that separations you get, very clear separations you get in the very estimates. But I'm just wondering, how how much sort of background work, if you will, do you need to have those fingerprints, those omic fingerprints, in order to identify those, or are those sort of readily available, you know, as markers of diatoms or what have you? Is that something that can be exported, or is that something that you'd have to catalog, develop your catalogs regionally?
Yeah, well, the answer is that that's the advantage of it, and you don't have to have that, because you're basically, you can't determine exactly what, unless you have that catalogue, a barcode of exactly the species, you can't determine exactly what it is, where the DNA has come from, but you can tell from what we know whether it's a bivalve or what genus, you know, lots of it goes down to that sort of genus and of that sort of information. And suppose the information generally left is say operational taxonomic units for these species. So, but you do know that you're losing all these types of polychaetes, or you're losing or increasing these numbers of polychaetes at these sites, you're losing these types of bivalves or ataxicoids and that sort of information. You know, you've got a huge amount of biofilms and things going on and so on. So that's the information you're getting. That's why I say it doesn't replace some of these other techniques. I know in some jurisdictions, Canada's going around barcoding all its streams, you know, things like that. So you know, people just go out there and collect the stuff, and you know if you're measuring that DNA, you've got exactly the species, you've got that information. China's actually doing it. They can do it quite easily because you've got the manpower, cheap manpower to have people do all the careful identification at the same time, do all the genomics of that side, the DNA, you know, barcoding information. So that's useful. But I think one of the big advantages of this is, you know, you go to a remote area, you know nothing. You know, you think about these species, the deep sea, where lots of people are chasing for mineral wealth or are heading that way. You know very little about the organisms that are present there. But you collect the DNA and go, well, these are definitely crustaceans, these are definitely bivalves, these are definitely, you know, these types of things, and using that sort of information in that manner.
What are you measuring the DNA in? Water, sediment, or
either anything?
Anything?
Yeah, any media. And you do have the contamination problems, that you can contaminate it with things. You've got to be careful in the laboratory as you do with any technique. But the sediment techniques, you can just take the sediment, and, you know, it's just a quite simple extraction techniques to get the DNA out. And basically, you've got everything. You've got, you know, our tree of life is us, and then there's all of the bigger eukaryotes and organisms that are like us, down to frogs and reptiles and other things, and then there's all the bacteria and then the archaea and things. And so you get everything if you want it, in that sense. Yeah.
A follow-on to Mark's question, I guess, is there a digital library that's available out there that we'd be able to?
Yeah, so generally yes, the answer is yes. That's where everything gets blasted against these big digital libraries.
Are they available
publicly? Yes. Yep. And basically you can't publish anything unless you put your data into these libraries. So nothing's publishable in this world unless you deposit all your gene information into these libraries. So they're getting better and better all the time as well in that way. And there's also, at the moment, say it's not the technique for us at the moment. There's a lot of informatics. So suddenly moving from time under the microscope to people creating the right pipelines to how do you process all this information and so on, you know, the storage for computers, the processing power for computers, you know, lots of A, T, Cs and Gs and things, that a lot of data to store.
Your DGT technique that you showed for metals, I think you mentioned that others are working on tablet templates for organics, but have you tried that particular technique on organics, or is it not suitable?
It's not suitable because it's for specific for binding cationic metals in this case, and the passive sample is specifically absorbing and accumulating, you know, certain organic contaminants and things. One of the, I suppose, come down to, you know, the simplicity of using these techniques, and the passive samples for organic will start to make it very good in many cases, but still, you don't see them being rolled out to assessments very much. There isn't the people going, you should be using a passive sampler, and I feel that needs to, you know, it does need to happen a lot more. People need to step to that side of going, you know, we need to start, you know, people develop these techniques, people just need to start applying them, even though there's a cost in doing so, to really, you know, prove them as how good they are as well. Mm-hmm. And so, yeah, further development on the middle ones, the sediments.
Those ones,
a whole range of.
For the DGT samplers, when you do field deployments, what do you think of as a typical deployment time? So what's the shortest time you put it out or the longest time to get an integrated measure?
Yeah. So in waters, they generally put them out for many days to 7, 10 days and things like that. And they can accumulate, and you've got very low concentration of water, and you can accumulate for that length of time. In sediments, generally you're only putting them out for 24 to 48 hours. So it's still not. Is it really a time integrated measurement? So over some period of time, and these experiments actually had several deployments of DGTs, and we're averaging and doing things in that sense, and they seem to be actually fairly similar, even though they're deployed at different times during these experiments. But really, for 24 hours, you go out there, you put them in, next day you take them out. But one of the challenges with passive samplers as well is it's hard to do passive sampling in many locations with people. People like playing with things, so you've got to have things where you can hide things and do things. And there's smart ways of doing that, buoys that disappear and so on. You're okay if you're at a mine site or an industry site where they control the whole area and they can do things, but often people tamper with things, so it becomes harder. Another aspect of time, I think about DGT too, and lots of these passive samplers, people go, you're disturbing the equilibrium. You've got to put them in so carefully, not introduce oxygen through sediments. And that's good if you want to really understand what the profiles of those metals were right then and at that period in time again. But an organism doesn't care about that. An organism's moving around, it's injecting stuff, it's pumping in oxygen. And so, to me, the ideal passive sample is probably one the size of an organism, and it moves around, does things, goes up and down, and that sort of thing. And so, the need to be that careful with how you put them in. It's probably better to put more in than actually worry about those stacks as disturbance occurs.
So how would this apply in an area where there's more dynamics and sediments? So there's, you're talking about a flux from depths where those depths are changing seasonally, storms. So how would you take the data and use it, or is it less usable in situations where it's much higher dynamics in the sediment?
I think the issue is that applies to all of the techniques for bioavailability. So regardless, sediments go anoxic, more so in summer when you get lots of breakdown of organic matter, and probably more oxic during winter when less is happening sometimes in different areas and so on. So there's always going to be those issues, and resistance. It depends what you're trying to protect as far as organisms and what is going on there. So I don't really see any differences compared to the other techniques. But no passive samplers or any of these bioavailability techniques get away from doing total contaminant analyses. Sometimes we go, we should just replace total contaminant analyses with some just to go directly to testing the bioavailability. But there's quite a big risk in doing that. In fact, that things change with time. So you've still got to know total concentrations. Have that in your mind that, well, things could become more bioavailable, but understand the contaminants in that sense. So,
another question. When you had plots of the DGT flux versus bioaccumulation, on some of the plots it looked like as the flux increased, the amount in the organism increased.
Yeah.
In other cases, for other metals, it looked like it leveled off. And then for another metal, I think maybe it was copper, it looked like it didn't go up much at all. Do you have any thoughts as to why? Well, first off, are you seeing different behavior for the different metals?
Yeah.
And do you have any thoughts on why that's occurring?
Yeah, and the answer is definitely yes. You see different behaviours for different metals, and it should be. Because organisms, copper's essential for organisms, zinc's essential for organisms, so that they're regulating and doing things differently. Mm-hmm. Some started with quite a high background of copper and zinc naturally, and therefore they've probably already got all their ligands and things in place for processing copper. So do they need to accumulate any more, or will they? And things like that. So it's a mixture of about bioavailability and about the physiology of the organism of what they're doing with those metals. Completely non-essential metals, things like cadmium, which is largely, not totally non-essential. Often just accumulate. They just go up.
So in the guidelines, you taking that into account by doing the normalization technique?
Yeah, and I find bioaccumulation hard to use, not in the guidelines, because there's no guideline for bioaccumulation.
There's a part where you normalize the DGT flux to the guideline, that's what I mean.
Yeah. As far as different rates of bioaccumulation, no, because I'm probably not using the bioaccumulation, but as far as the toxicity, yes, probably related to the toxicity. When I'm looking at bioaccumulation, we're only looking at one metal at a time, so not looking at them in a sense of having them all clustered together. Bioaccumulation data is hard to use for metals because many of these metals are essential. If you take an organism and go, someone's got out in their field, collected these sediments from the Hudson River, these organisms, the Hudson River, whatever they are, depurated them, sent them to the laboratory and gone, well, the concentrations are high, and you go, well, they've been there a long time, they've been accumulating for a long time, doesn't mean there's a risk to the organism itself. You know, the accumulation is hard to use because if they're processing and storing it, there are whole ways of storing things inertly in organisms in different parts of their body, and you measure that as a concentration. You know, so if you need to understand what that, you know, what's the flux then and what's the intensity of some organism, and you can't do that from bioaccumulation. Organics is different. You know, organics act differently in many cases, and therefore the accumulated concentration is far more useful for organics than it is for metals. So sort of there's been a lot of misuse of metals data in that sense, and very hard to use.
I have another question. Can you just explain a little bit when in the flux calculation of more of a practical detail? So I think your flux is a time factor in flux, so it's a per time or rate. But there's also a depth component accumulation. So are you with the plot that you're showing there? Are you showing different time? Or it seems like you cut you took sections of the different of the tool length.
Yeah.
So it seems like it was related to depth, but then it's related to time. Can you explain that?
Yeah. So flux is related to an area. So you've got a flux through a surface or an area, and basically we're having a flux through a certain area on our DGT probe. So we're slicing it and taking that area, and at the centre of that area is what we're showing the flux is. So it's the average over that area in space. So it does have, you can get the flux at that depth or that depth, but it's representing an area in that space. So it's, you know, micrograms of copper or micrograms of metals are moving per time and such. And it's similar, you know, what you have out of a sediment into the overlying water, you know, it's micrograms per area of sediment per time.
Can I assume on the plot that those are standardized areas based on the selections that were taken?
Yes.
And then the plot is versus depth for a given time?
Yeah, that's right. So that, yeah, then you've got a flux. Yeah, so the flux is occurring at a higher rate. Copper in word space, iron and manganese and things deeper down, and other metals at different areas too. It's interesting. Something else I didn't talk about here. Sorry, going on is, you know, many of these organisms, dissolved exposure isn't the main exposure. This is when you're doing things in the laboratory and you dose them with high dissolved concentrations. But in many environments, dietary is their main route of exposure. I mean, these organisms have to eat, and they're not necessarily just picking off the clean algae and the clean amphipod. They're getting a lot of sediment with them along the way. And virtually all the literature you see and the work we've done shows that, you know, in many cases, it's a greater proportion of exposure comes from what they eat and diet. And sometimes we go, well, how does flux, which is that dissolved exposure, relate to toxicity? I'm saying, in my amphipod particularly, most of that exposure comes from diet. And it's confusing for some time, but I think in many cases with these contaminated sediments, your source of dissolved contaminants is from the particular place. There's a flux out from these sediments, and it's just the fact that you've got a very good correlation between what's on the particle in many of these contaminated sites and what's going into the pore water, into the overlying water. And so, you know, you predict your toxicity based on what's in the pore water, but it's actually a combination of what's in the pore water and what you're eating and all these things. You know.
I'd like to ask, do you also study uranium in these sediments?
in these sediments, not specifically, because the concentrations are low, but Australia's got a lot of uranium. In our freshwater well, I've done studies with uranium, more so, not in our coastal marine well. You know, we've looked at it, but there's nothing there to make it go into the ranges of concern, certainly downstream of our uranium mines in Kakadu National Park, some issues, closing mines and having no closure criteria at all. But then once again, that uranium tends to be cationic, but it's also an oxyanion as well. So you need there's actually a uranium DGT, say, it's metazole, but it's a different binding layer that's quite specific to uranium. And been shown to be quite useful. Once again, nobody's actually picked it up as industry to start using it. It's a really good technique. Nobody's using that probe yet. People actually developed them now for ammonia, so there's actually a nutrients DGT developed with arsenic, selenium, different binding phases and things, and people can mix these binding layers together, so you actually accumulate they're all together, lots of different things. So there's becoming smarter ways of getting lots of information.
Just one overall question: does the regulatory environment are they guiding research in any way? Like, are the regulators more interested in metals and sediments, or do you have organic contaminant issues that are plaguing Australia, or what's driving the direction of a lot of your research?
Yeah, I'd say the contamination issues are fairly broad. I mean, we've got certainly huge areas of docks and PCBs and things like that in Sydney Harbour. You don't catch fish or eat fish caught west of the bridge, parts of high human consumption. So there's those sort of issues. You've got metal contamination everywhere, but it's very specific. Out west into our arid inland, it's all about metals. It's more of the mining industries. There's no big other types of petrochemical, other sort of industries inland, even though we're starting to frack everything as well in Queensland and so on. So it's fairly broad. Is the regulator driving it? Australia's a small country. I made that comment before. We're smaller than population. As big as the United States, but our population is small as smaller than California. And so we're a smaller scientific community, and the regulators are smaller, and there's still fractured. There's the federal, and then there's all the states doing their own little things. But some degree, the science that we provide is a smaller community. Is generally more accepted, so it's easier to get policy and things. Momentum's slow, but it's still faster than what happened here with things. I think about when we're the US, when we're guidelines developed for waters in the states, and when are they going to be revised, or when are they to get revised, or what? How do you implement a guideline? Say actually we need to put some numbers in place. I was having this discussion with the USEPA with the super fund office. It's actually, I don't necessarily have numbers they can enforce, but it's quite useful having a number to go, right, this is a number that all industry and everyone has to think about. That we think this is too high, and then you've got to start arguing, discussing, and doing science around those guideline numbers, despite their uncertainty.
You have a question? Okay. Nobody online? No. Okay, well, Stuart, thank you. Reminder, we have another seminar next Tuesday. So thanks. If you haven't signed in, please. Where's the sheet? Is the sheet up?
The sheet should be out front, and please RSVP so we know how many people are coming and we can set up the room appropriately. Thank you.
Thank you.